Welcome to G Fun Facts Online, where we dive into the most amazing science happening right now. Imagine a world where we can map the biodiversity of an entire forest, detect a super rare endangered animal, track how agricultural diseases are spreading, or even sniff out illicit substances, all by just taking a sample of the air around us. Sounds like something straight out of a sci-fi movie, right? Well, buckle up, because this isn't fiction. It's the rapidly advancing field of airborne environmental DNA analysis, or eDNA for short. This technology is poised to completely revolutionize how we monitor the living world and even human activities. So what exactly is this eDNA? Think of it as tiny genetic breadcrumbs. Every living thing, plants, animals, microbes, even us humans, constantly sheds genetic material into the environment. This could be through skin cells, hair, saliva, pollen, spores, or waste products. For years, scientists have been fishing this eDNA out of water and soil samples to understand aquatic life, or check out what microbes are chilling in the dirt. But now, this incredible tool is taking to the skies. Researchers are discovering that the air itself is like a massive library of genetic information, carrying these tiny fragments of DNA from a huge array of life forms, from the smallest microbes to the biggest mammals, and even traces of stuff related to us humans. The best part? It's non-invasive. We can learn so much with minimal disturbance to the environment. Okay, let's talk about how airborne eDNA is changing the game for ecological monitoring. The applications here are massive and super exciting. First up, comprehensive biodiversity snapshots. Imagine taking an air sample and getting a list of almost everything living in that area. Studies have successfully identified DNA from tons of plants, mammals, birds, amphibians, reptiles, insects, fungi, and bacteria, all from a puff of air. This is incredibly powerful, especially in places that are tough to survey normally, like dense jungles or remote mountaintops. It's also great for tracking those shy, elusive, or nocturnal critters that are hard to spot. Get this. In some studies, airborne eDNA surveys found more species with less effort than traditional methods like visual counts or trapping. For instance, researchers at a zoo were able to identify a whole range of their captive animals just from air samples, and even picked up DNA from wild animals living nearby, like the endangered Eurasian hedgehog chilling outside a UK zoo. How cool is that? Next, think of it as an early warning system for threats. This is a big one. Airborne eDNA can act like a smoke detector for ecological dangers. It can spot invasive species before they really take hold and cause massive damage. In farming and forestry, it can detect the airborne spores of plant diseases before the crops even look sick. This means farmers can target their treatments precisely, potentially cutting down on the need for broad-spectrum pesticides. There's even a technology called AirSec that literally sucks in air to find DNA fragments of disease-causing fungi, helping farmers fight crop diseases much more effectively. And it doesn't stop there. Airborne eDNA helps us in tracking ecosystem health and environmental change. Because the air is always changing, so is the eDNA in it. This means we can track shifts in plant and animal communities over time. Are biodiversity levels changing due to climate change? What's the impact of pollution? Are habitat restoration efforts actually working? Airborne eDNA can provide the data. For example, studies have shown that restoring rangeland can significantly change the detectable airborne eDNA of the plants in that area. Scientists are even looking at old air pollution filters, some from decades ago, and analyzing the eDNA trapped in them. It's like a genetic time capsule, giving us a historical look at biodiversity changes. Even in big cities like London, airborne eDNA is being used to monitor air pollution, seasonal pollen levels, fungal spores, and even genes related to antimicrobial resistance. This data can help with public health and urban planning. Beyond spotting pandas and pathogens, the sensitivity of airborne eDNA is opening up entirely new frontiers, especially in forensic science and monitoring illicit substances. Imagine detecting contraband from the air. Believe it or not, researchers have successfully detected the DNA of illicit plants, like cannabis and poppies, which are used to make opiates, just by analyzing the air in urban areas. This hints at future applications for monitoring drug production or figuring out consumption hotspots. This tech could also be a game changer in fighting the illegal wildlife trade. Imagine being able to detect DNA from trafficked animals, or their products in transit or at checkpoints, just from the air. Then there's forensic investigations. The idea of capturing human DNA from the air is also being explored for crime scenes. Airborne eDNA could potentially offer non-invasive biological evidence. If someone was present at a scene, they might have left traces of their DNA floating in the air, even if they didn't touch anything. This could be super useful in cases where there's very little other evidence. And here's a really interesting one: the pharma biome. This is the eDNA found on or inside fake medicines. Scientists are investigating if this can be used to trace where counterfeit drugs are made, which would be a huge help in combating a serious global health problem. So how does this all actually work? How do scientists grab these invisible clues from the air and make sense of them? It starts with collecting the invisible clues. Airborne eDNA is typically collected using either passive or active samplers. Passive samplers are pretty simple, like dust traps, and they just rely on the wind to deposit particles. Active samplers are more, well, active. They use pumps or fans to draw a specific volume of air through a filter or into a collection liquid. How much DNA you get can depend on things like how much air is sampled, for how long, and how fast the air is pulled through. Researchers are constantly cooking up new sampling devices, even ones that can be deployed by aircraft for surveys high up in the atmosphere or over vast areas. Some studies even suggest that for long-term, unsupervised monitoring, those simple passive samplers might actually be better and more cost-effective. Once the samples are collected, it's time for decoding the genetic data. The genetic material is first extracted from the filters or the liquid. Then, techniques like polymerase chain reaction, or PCR, you might have heard of it, are used to make many copies of specific DNA regions. This is like hitting Ctrl+C, Ctrl+V on the DNA you're interested in. After that, next-generation sequencing, or NGS, allows scientists to read millions of DNA fragments all at once. A really key approach in eDNA studies is something called meta-barcoding. This lets scientists identify multiple species from a single complex sample, like our air sample. They do this by targeting specific, standardized gene regions. Think of them as genetic barcodes that are different between species. And as sequencing technology gets even better, with things like long-read sequencing, the accuracy and detail of these analyses are just improving all the time. Now, as amazing as all this sounds, airborne eDNA technology is still pretty new, and there are definitely some challenges and hurdles that researchers are working hard to overcome. Let's talk technical hurdles. One biggie is DNA degradation. DNA floating around in the air is exposed to things like UV radiation from the sun and temperature changes, which can break it down. This makes it harder to detect and identify. Sample contamination is another critical concern. We're often dealing with tiny amounts of DNA, so preventing contamination from other sources, including the researchers themselves, during collection and analysis is super important. Scientists are also still figuring out the best ways to collect DNA efficiently, especially in environments where there isn't much life or when they're looking for a really rare species. Interpreting the data also has its complexities. If you detect DNA, does it mean the organism is there right now? Or was it there a while ago and its DNA is just lingering? It can be tricky to tell. Plus, figuring out if a lot of eDNA means a lot of animals, or just one very sheddy animal, isn't straightforward yet, though it's a hot area of research. Understanding how far airborne eDNA can travel from its source, and how long it sticks around in different atmospheric conditions, is also crucial for making sense of the results. Then there are the analytical and logistical challenges. The accuracy of identifying species heavily depends on having really good, complete genetic reference databases. If a detected DNA sequence isn't in the database, you can't identify what it came from. Choosing the right DNA markers, those gene regions we talked about, and the primers used in PCR is also critical, because different markers work better for different groups of organisms. And finally, the computational tools used to process and analyze all that sequencing data, called bioinformatic pipelines, have to be designed very carefully to avoid errors and biases. This next point is a really important one: ethics. A significant ethical consideration in airborne eDNA research is the accidental collection of human DNA. Scientists call this human genetic bycatch. Since we humans are constantly shedding our DNA into the environment, air samples will inevitably contain our genetic material. Now, this could have some useful forensic or medical applications, as we discussed. But it also raises serious privacy concerns. Who owns that airborne human DNA? How should it be handled? There's a growing discussion about the need for clear ethical guidelines and regulations for collecting, analyzing, storing, and using human eDNA to protect individual privacy and prevent any misuse of this sensitive genetic information. It's all about balancing the quest for scientific breakthroughs with protecting human rights, and that's a conversation we definitely need to be having as this technology develops. So what does the future hold for airborne eDNA? Well, it's incredibly bright, and the field is evolving super fast. Scientists are working flat out to improve the sensitivity and accuracy of detection methods, making it possible to identify even the faintest genetic traces. Imagine portable, field-ready devices that can do real-time eDNA analysis on the spot. This could totally revolutionize how we respond to things like pathogen outbreaks or new invasive species. There's also huge potential for integrating airborne eDNA sampling into existing large-scale monitoring networks. Think about all those air quality monitoring stations around the world. What if they could also become biodiversity observatories, constantly providing data on ecological health? That would be amazing. The applications are expected to expand even further. We're talking public health, like tracking airborne allergens or viral pathogens. Biosecurity, monitoring for bioterrorism agents or agricultural pests. And, of course, enhancing conservation strategies for our planet's wildlife worldwide. Airborne eDNA analysis is way more than just a cool new scientific trick. It really represents a paradigm shift in our ability to see, understand, and interact with the biological world, and even the traces we humans leave within it. As this technology continues to mature and scientists tackle the challenges, the sky is truly the limit for understanding and safeguarding our planet's precious biodiversity, and potentially for creating safer and more informed human environments for all of us. Who knew there was so much information just floating in the breeze? That's all the time we have for today on G Fun Facts Online. Thanks for tuning in, and I hope you're as excited about the future of airborne eDNA as I am. Until next time, keep looking up and keep wondering.